Hi everyone, I'm Ons Masmoudi, PhD student from Institute Pasteur of Tunis. I'm glad to be here today to present to you my work entitled Leveraging Data Balancing and Chemical Encoding Strategy for Robust AI-Based Drug Discovery Pipeline. So we will start with an introduction, presenting our methodology and our result, and we will end up with a conclusion. So the emergence of AI have brought a major advancement across various fields. And here we explore how AI is making a significant impact on the drug discovery field. Drug discovery has been traditionally very expensive and very lengthy process. So AI is revolutionizing this by predicting the properties and activities of chemical compounds with low cost and rapidly, or the opportunity to generate novel compounds. So here we present three main scenarios where AI can have a significant impact across the different stages of the drug discovery. And in our groups, we focus on the target to hit and hit to late stage. However, the success of an AI drug-based pipeline relies heavily on large and high-quality chemical dataset for training. But unfortunately, the available dataset suffer from class imbalance, in which a few molecules are active and most of them are inactive. And this will make our models based toward the majority class, and our classification will be difficult. Another key challenge lies in encoding the chemical structure. So a molecule is defined as a group of two or more atoms connected by chemical bonds. Our hypothesis is that the chemical structure is not a random arrangement, but it is the underlying framework of the pharmacological activity. That means the spatial arrangement of atoms within a molecule will make a creation of chemical groups that are responsible of the functionality and the pharmacological activity of molecules against a specific target. And this step is challenging to artificial intelligence model to work with this data. That's why we have to encode it through different representation like fingerprint, molecular graph, etc. So in our work, the main objective is to optimize the dataset characteristic, including the class imbalance and the content, optimizing the encoding system and the predictive model in order to obtain a robust and real-world AI-driven drug discovery pipeline for predicting the biological activities of chemical compounds. So as a methodology, we followed a standard pipeline of an AI-based project. So first we collected our data, then we encoded our data into different representation. So our data comes in format of smiles, which is a textual representation. So for machine learning model, we use the molecular fingerprint, which are binary vector that encode the absence or the presence of certain substructure. For graph neural network, we encode our data into molecular graph, which is the molecule will be presented as an indirect graph where the nodes represent the atoms and the edge represent the bonds. And for transformers-based model, we tokenized our smiles into discrete tokens. Then we will split our data into training validation test set, and we will assess the performances of our models. So this pipeline was applied to 4 different data sets which have different imbalance ratio. And to assess or to see how to, to address this challenge of class imbalance, we use different resampling methods, which are the standard methods of resampling, like random oversampling, smooth, random undersampling, near miss, and we propose a novel strategy called K-ratio undersampling, in which we generate 3 different ratios, which are 15, 20 to 25, and 10 ratio. And we assess the performances of our models under the different configuration based on several metrics, based on the confusion metrics. So moving on the results part, and first we evaluate the model's performances when trained on the original and the standard resampling techniques. And here we demonstrated that the random undersampling was more effective than the random oversampling techniques across several metrics, and when comparing the 8 model under the 5 data states and across the 4 diseases. However, in other studies oversampling techniques was more effective in handling the class imbalance. But in our case, they are not suitable to our chemical data. And random undersampling was more effective despite the loss of information. So the critical question now, to which extent the imbalance ratio is hindering the model's performances, and is there a satisfying imbalance ratio where the model's performances will be improved? So that's why here we will compare the performances of each model on the different data states, and we observed that when the class imbalance is decreasing, the performances of the models is increasing and showed its best enhancement under the 10 RAS configuration. However, only the GAT model did not improve across the different data states. So through the radar plot here, we provide a visual summary of the model's performances through the F1 score metrics for each dataset and across the various imbalance ratio and setting. And here we select the top performing model for each dataset, and subsequently the selected model went through an hyperparameter tuning and a cross-validation step. So here, up to now, we observed that the HIV dataset led always to a worst value, and they are not satisfactory. And as we see in this table, each dataset presents a different top-performing model, and here we should tailor the model selection to the unique characteristic and the specific challenges posed by each dataset, which means there is no universal model that can work well with all the datasets and all the cases. So through the hyperparameter tuning, we noticed a notable improvement in some models' performances like MLP, random forest, and more former, as observed in the table. And here we highlight that the hyperparameter tuning and the cross-validation are critical steps in optimizing the model's performances. So now we performed an external validation to the optimized model, which involved the use of 4 confirmatory DIUSA linked to the primary dataset analyzed up to now. The aim of this step is to assess the generalization power of our models to identify the biological activity on unseen data, and we will assess that our model can be used in real case data. So for each model, we compute the true positive rate and the false positive rate, and we will evaluate the trade-off between both metrics. So here, we evaluate the top-performing model under 3 different configurations, which are original, 1 to 10, and random undersampling. And as we see here, the triangle shape, which represents the 1 to 10 RAS, is on the targeted zone. So here we aim to optimize the trade-off, which means we have to maximize the TPR and minimize the FPR. And taking the example of the malaria dataset, where we see that almost all the models perform well under all the state, but 1 to 10 strategy achieved the best balance between true positive and false positive rate. And here also we demonstrated that the HIV dataset is very challenging, as it doesn't represent models that attain the targeted square. So that's why, so up to now, we confirmed that achieving a MELD imbalance ratio through KRAS led to the best performances, while we balanced the trade-off between reducing the class imbalance and preserving the data integrity. And for the drug discovery task, optimizing the trade-off between TPR and FPR is highly insightful to identify more active molecule and minimize the false positive, which is crucial to the success rate of the drug discovery outcomes. And now, up to now, we demonstrated that each dataset presents unique challenges primarily due to the chemical diversity. That's why we will conduct an intersect analysis of the dataset content. So here we will compute the Tanimoto similarity between compounds coming from the training dataset and the confirmatory dataset. And then we will use a chemical space network to see the relationship between both between molecules in both classes and both datasets. So for the most challenging dataset, which is the HIV, for the active molecules in the external test set and the inactive molecules in the training set, we observed a densely interconnected chemical space network, which means the presence of high number of molecule similarity between both classes. And this explains the high rate of false negative in which we have low TPR. So here the models will not be able to distinguish between both classes. However, when we see the inactive molecules in the external test set versus the active molecules in the training set, we observe they don't have there are not too much molecules that have a similarity between both between them, and this is confirmed by the absence of false positive and the low FPR. For the best case study, which is the malaria dataset, we observed a structural diversity between active and inactive class, which contribute to more accurate prediction as observed in the external validation section. As a conclusion, this study proposed an optimized pipeline for an AI-based predictive framework for drug discovery working on high-throughput screening dataset. And we highlight that addressing the class imbalance and optimizing the data content to improve AI-driven drug pipeline is very crucial, and the 10RAS strategy significantly improved the model's performances. And as a perspective, we aim to automate the optimized pathogen-specific pipelines toward their deployment in our open-source platform named CDALSDB for use by the scientific community. So you can scan the QR code to access to our platform, and now it is available to have the prediction of two pathogens, which are the coronaviruses and the leishmaniasis. So I want to thank too much my supervisor and especially Dr. Emna Hriga for her support and guidance. So if you have any question, you can contact us. Our work now is under revision, and you can follow us on the Pine Project LinkedIn page to have news about our work and our progress. Thank you very much.
Any questions?
A fantastic presentation. I was smiling all along when you were presenting. And I think you answered one of my questions towards the end in terms of looking at the structural activity landscape of the data set that explained why you were seeing those performances in the predictions. The other thing that I might also want to call your attention to is the target specificity of the compound in the data set. So to have a malaria data set, the question now is, what is each compound targeted in terms of mechanism of action? That can also be a confounding issue in terms of the noise in the prediction. Thank you.
Do you want to comment? Or any other questions?
Can you comment?
Yeah.
Well, thank you for your comment. Actually, it's the approach we have here is not for biologists. While I'm responding, there is no targets involved in our approach. It's basically a linked-based approach that doesn't really take into account the molecular targets, which basically may be confusing for some classical approaches. But still, it helped us have a very large overview of what is working on the parasite itself, without any type of information about the targets, which is very important, but can obviously be, deconvoluted afterwards once we have novel molecules identified.
Stop share. Thank you.
Thank you.
So if